Hi friends, wishing you the very best for your exam. In this video, we have a quick paragraph-wise revision of chapter Biotechnology Principles and Processes. Without wasting a second, let's begin. The first paragraph tells us about what is biotechnology. There are two aspects of biotechnology. The first one is called the traditional biotechnology, where we are using these organisms from the time immemorial in the making of curd, then preparation of wine, brewery, in flavoring, etc. We are using yeast and other microorganisms. In traditional biotechnology, we are using the innate capacity of microorganisms without making any change in microorganisms. Whereas there is a second aspect of biotechnology, which is modern biotechnology, where we are editing the genes or incorporating different genes into different organisms for getting some desirable traits. Classical example is this humulin, that is recombinant insulin that is produced inside bacteria. We have incorporated insulin gene into the bacteria. Bacteria just acts like a biofactories for the production of insulin. Then this is vitamin A rich golden rice, where a gene is incorporated that helps this crop to produce vitamin A. Then this is flavor saver tomato, a genetically modified tomato that has increased shelf life by suppressing or silencing the ripening gene. So, in all these examples, we have changed or modified the genes or DNA. That makes the modern biotechnology. So, according to European Federation of Biotechnology, it defines biotechnology encompasses both traditional and modern molecular biotechnology. So, the integration of natural science and organisms, cells or parts, for the production of goods and services. The second paragraph deals about two core concepts in biotechnology. The first one is genetic engineering. That means we are editing the gene. We may be incorporating a gene or silencing a gene or editing a gene for getting a desirable trait. The second aspect is bioprocessing engineering. Bioprocessing engineering means providing a sterile condition for the growth of microorganisms or an eukaryotic cell to produce a particular product in large quantities, like this bioreactor, for the production of antibiotics, vaccines, enzymes, etc. The next paragraph is about the difference between hybridization and recombinant DNA technology. So we have all these crop varieties by means of hybridization, both animal breeding and plant breeding. So we have crops with desirable traits. So what's the basic difference between hybridization and recombinant DNA technology? While hybridization will be getting some desirable qualities, but undesirable genes are also transferred along with the desired genes. But in recombinant DNA technology or gene cloning, we are specifically isolating and introducing a set of desirable genes without introducing undesirable genes into the target organism. So the process is specific. We are introducing a specific gene that will provide a desirable trait in the host. So that's the basic difference between hybridization and recombinant DNA technology. Then the next paragraph deals with how a foreign DNA replicates inside the host. For replicating inside the host, there is a sequence required that is called as ORI, or origin of replication. So suppose this is a bacterium, so this is an ORI, origin of replication. This sequence is a must for replicating inside the bacterium. So any gene that is attached to this ORI can replicate inside the host. So that's the concept. For cloning, we can join an ORI with our desired gene of interest to get it replicate inside the host. The next paragraph deals with the development of first recombinant DNA molecule by linking a plasmid with an antibiotic-resistant gene that is isolated from Salmonella typhimurium, and it was done by Stanley Cohen and Herbert Boyer in 1972. So this was the procedure. So plasmid, as you see, this is the plasmid. So plasmids are extrachromosomal double-stranded circular DNA molecule that is present in bacteria. So they have isolated a plasmid from Salmonella, then incorporated an antibiotic resistance gene. So these two are joined to form the recombinant DNA molecule. So DNA from two sources, that is why called as recombinant DNA molecule. So let me make it more clear. In the next paragraph, the tools used for making the recombinant DNA molecule, the first one is restriction enzymes. They are called as molecular scissors as they are used to cut DNA fragments. So suppose this is the antibiotic resistance gene and this is a plasmid from Salmonella. So we have made a cut into this plasmid using restriction enzyme. then incorporated our antibiotic resistance gene to this plasmid. So we have a recombinant DNA molecule. Then another enzyme is also involved, the joining enzyme, which is called as DNA ligase. So this is joined by means of DNA ligase. These two DNA fragments are joined by DNA ligase. Now we have a recombinant DNA molecule. So this recombinant DNA molecule, as plasmid is naturally present within the bacterium, it can self-replicate inside bacterium to produce multiple copies of that antibiotic resistance gene. Basic steps involved identification of DNA with desirable genes, introduction into a host, and maintenance of that DNA within the host, and multiplication of that particular DNA. Then details about the tools used in recombinant DNA technology. The first one is restriction enzymes. So restriction enzymes and methalase. So two enzymes that were isolated from E. coli, and restriction enzymes were later called as restriction endonuclease. The first restriction enzyme isolated was Hin2 from Haemophilus influenzae. Now let us see, now we have almost 900 restriction enzymes isolated from our 230 strains. Now let us see how this restriction enzyme is named. The classical example is EcoR1. As you see, it is derived from bacteria Escherichia coli. So the first letter of genus name E and the second two letters of species name CO, Eco. R is the strain name, and whereas this one indicates the order in which the enzymes are isolated from this particular strain of bacteria. So this is how EcoR1 or restriction enzymes are named. In the next paragraph, it is about the restriction enzymes. There are two types of restriction enzymes, exonucleases and endonucleases. As the word suggests, exonucleases makes terminal cuts or cuts from the ends, as you see here, whereas endonucleases can make cuts within DNA molecule. The next is regarding the restriction sites. These restriction enzymes, especially type 2 restriction enzymes, can cut at specific sites called restriction sites. So these are often palindromic sequences. So let us see this EcoR1. As you see, the restriction site is GAATTC. So vector DNA is cut at GAATTC. Foreign DNA is also cut at GAATTC. So we'll be getting the fragment like this. So this is called sticky end. As you see, there are single-stranded unpaired regions. So here, if there is a complementary strand, it can easily form hydrogen bond. That's why it is called a sticky end. So EcoR1 makes a sticky end. So if this vector DNA and foreign DNA is cut with the same restriction enzyme, we can, if it is sticky ends, it forms sticky end, we can easily join to form recombinant DNA molecule. As this complementary base pairing happens, so it will easily form hydrogen bond if there is a complementary sequence. The next point is regarding the restriction site. So restriction sites are palindromic sequences. As you see, let's take the same example, this EcoR1. As you see, 5', 3', it is GAATTC. So from here, 5', 3', you can see it is GAATTC. So these are palindromic sequences. Read the same from opposite direction. So from this direction, it is GAATTC. From here also, it is GAATTC. That's why called as palindromic sequences. So we have already discussed what is the sticky end. Sticky end means restriction enzyme that makes cuts that will form single-stranded unpaired regions so that a complementary sequence can easily form hydrogen bonds to make recombinant DNA molecule. So restriction enzymes that produce sticky ends are preferred in recombinant DNA technology. So this is a summary of the entire process. So foreign DNA, then vector DNA. So both these are cut with the same restriction enzyme. As you see, a cut is made. So here also the plasmid is cut with the restriction enzyme. Then this is joined. So maybe this produces sticky ends like this. So you can see these are the sticky ends and these are the sticky ends. So annealing is very easy by complementary base pairing. It forms hydrogen bond. So annealing is very easy to form a recombinant DNA molecule. Recombinant DNA molecule is so called because this DNA is from two sources. This is plasmid DNA from bacterium and this is the gene of interest from any source. So we have a recombinant DNA molecule and this recombinant DNA molecule is introduced into the host. Here it is E. coli or bacterium. The process is called as transformation. Introduction of recombinant DNA into the host is called as transformation. Then this cell divides, thereby replicating the copies of the desired gene. Also, this plasmid can replicate inside this bacterium. thereby also increasing the number of copies of that particular gene. In the next paragraph, it is about the separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease results in the formation of fragments, and these DNA fragments are separated by gel electrophoresis. Electrophoresis, as you see, DNA is negatively charged because of the phosphate group. So if DNA is loaded here, it will move towards the positive electrode. So the smaller fragments will move fast, reaching the bottom first, whereas larger fragments remain at the top. So this agar is isolated from seaweeds. It is just like a sieve that sieves DNA based on size and charge. So the smaller fragments reaches the bottom first compared to larger fragments. The next step, the separated DNA fragments are visualized using ethidium bromide stain. Pure DNA fragments is not visible under visible light. So we'll be staining with ethidium bromide. So this is ethidium bromide molecule. It will bind to DNA. It is a DNA intercalator. So we can see orange colored bands of DNA if this ethidium bromide stained gel, as you see, is exposed to UV fluorescence. So this is the DNA fragments, or bright orange colored bands are formed when stained with ethidium bromide when exposed to UV light. Then these separated DNA bands can be cut out. The process is from the agarose gel, and the process is called as elution. So this is elution, and these DNA fragments can be purified and can be further used for the construction of recombinant DNA. Then the next paragraph is regarding the cloning vector. Cloning vector, as you see, any DNA molecule that can carry our gene of interest is called a cloning vector. Classical examples include plasmids and bacteriophages. They can replicate inside the bacterial cell independently. As plasmids are naturally present in bacteria, bacteriophages normally infects bacteria or viruses that attacks bacteria. So they are present in high copy number per cell, so widely used as vectors. So plasmids with high copy number can be used as vectors, or that is present in multiple copies that can be used as vectors. Common vectors includes this plasmids, then bacteriophage. Bacteriophages are viruses that attacks bacteria. So if we are incorporating our gene of interest into this bacteriophage, they can naturally infect the host and transfer this gene. So we can genetically engineer this plasmid for transferring our gene of interest. The next is regarding the qualities of a gene cloning vector. So let me make it more clear with the simple examples. So what are the minimum requirements of a gene cloning vector? So this is a gene cloning vector which is genetically engineered, which is called as PBR322. As you see, it should have an ORI. Origin of replication is a sequence that is essential for replicating inside the host. You should have selectable markers like ampicillin resistance gene, tetracycline resistance gene to select the transformed recombinant cells. The third, it should have multiple restriction sites, unique restriction sites. And the fourth most important thing, this restriction sites should be within this selectable markers. So this helps in selection of transformed recombinant cells by insertional inactivation. At the end of a transformation experiment, we'll be getting three types of cells. The first one is non-transformed. Majority will be non-transformed. Nothing happened. Vector hasn't been introduced. So the second one is transformed with non-recombinant vector. So the vector is there, but our gene of interest is not there. The third one is transformed with recombinant vector. So vector with our gene of interest. So this is the colonies that we need to select. Suppose we have introduced our gene of interest in tetracycline resistance gene. So once we have introduced our gene of interest into the tetracycline resistance gene, this gene is no more functional. So that means non-transformed. So while we grow these colonies in tetracycline or ampicillin containing medium, both tetracycline and ampicillin containing medium, this non-transformed cannot grow as it doesn't have antibiotic resistance gene. But this transformed with non-recombinant vector can grow in both. As you see, this is ampicillin resistance gene and this is tetracycline resistance gene. Both are intact. So this colony can grow in both ampicillin resistant medium and tetracycline resistance medium. The third one, that is transformed with recombinant vector, can grow only in ampicillin resistance medium as this gene is intact. We have introduced our gene of interest in tetracycline region, so this gene is no more active. This is inactive. The process is called as insertional inactivation. Inactivation of a gene upon inserting another gene within the sequence. So that transformed with recombinant vector These colonies can grow only in ampicillin medium, cannot grow in tetracycline medium. This is how we select our transformed with recombinant vector colonies. That is why these genes are called a selectable marker, or that helps in selection of transformed recombinant colonies. Hope you are clear. So you can watch our video for more on this topic. The link will be in the description. The next is, we have already discussed how antibiotic resistance genes helps in selection of recombinants. Then the next is, one more point is added in this paragraph, and that is about beta-galactosidase gene used as selectable marker. So we have discussed how antibiotic resistance gene is used as a selectable marker. In beta-galactosidase gene, a chromogenic substrate if added, this gene if present will yield a blue color. Let me take the pen. So gene of interest is inserted within beta-galactosidase gene. So transformed recombinant ones, no blue color due to insertional inactivation. The colonies will appear white. So this is the colonies that we want. Whereas non-transformed colonies appears blue because the beta-galactosidase gene is intact. The process of inactivation of a gene upon insertion of another gene is called as insertional inactivation, so that we can easily select our transformed recombinant colonies. Now regarding the vectors for gene cloning in plants and animals. In plants, the most common and the most effective one is Acrobacterium tumefaciens, a pathogen of several dicot plants. It's a natural genetic engineer. It has a DNA plasmid called as tDNA, T plasmid. T plasmid means tumor-inducing plasmid, so which has a DNA which is called as tumor-inducing DNA. And naturally, this plant can incorporate this DNA into the plant genome, and this DNA has auxin and cytokinin genes that cause division of cells in plants, causing crown gall disease. And plants will synthesize certain chemicals that boost the growth of this Acrobacterium tumefaciens. So what we do is, we will be genetically engineering this tDNA. Our gene of interest is incorporated in this place so that this Acrobacterium will transfer that gene into the plants with high efficiency, as it is a natural process. So tumor-inducing plasmid is used that is modified into a gene cloning vector, widely used in delivering the gene of interest in plants. Similarly, retroviruses are also used in animals for delivering genes. So retroviruses DNA fragments are ligated into suitable vector, and that is transferred into bacterial, plant or animal host, where it multiplies and transfers our gene of interest. The next is how these cells are made competent or make it more suitable to accept the foreign DNA for transformation. So once suppose this is a bacterium, so this DNA should be transformed inside the host. How this is possible? As we know, DNA is hydrophilic molecule, so it cannot pass through the cell membrane. So we need to make it competent. The ability of a host to accept DNA is called as competency. So we have to increase the competency. This is by treating with many chemicals like divalent ions such as calcium, so that makes the cell more permeable, creating pores in the cell wall. Then there is heat shock mechanism. As you see, recombinant DNA is forced by incubating cells in ice, followed by heat shock at 42 degrees Celsius, and quickly putting back them on ice. This enables bacteria to take up DNA. So this is one method, heat shock mechanism. First one is provide calcium ion treatment. Then third one is micro-injection. By using micro needles, micro manipulators, we are directly injecting the DNA into the nucleus of animal cell, as you see here. Then one more method that is very suitable for plants because of the cell wall, tough cell wall, is biologistic method or gene gun method. DNA is coated with gold particles and forcefully bombarded with high velocity. This coated DNA is injected into the plant cell. So this is a method then that cell is grown in tissue culture, finally forming a transformed plant, transgenic plant. So this biologistic method is widely used in the case of plant cells because of its tough cell wall made up of cellulose. Then disarmed pathogen vectors are used. Disarmed means the pathogenic capacity, the gene that is causing pathogenicity is replaced with our gene of interest, and that is transferred into the host. Then the next part is regarding the process of recombinant technology, DNA technology. This is a summary. Isolation of DNA, fragmentation of DNA using restriction endonuclease, then isolation of desired DNA fragment by elution, then ligation of DNA fragment into a suitable vector, transferring into a host which is called as transformation, and finally culturing of host cells in a medium for multiplication or expression. Then isolation of genetic material, the processes involved this is a summary. DNA isolation, it is depending on the organism. The cell wall lysis should happen. In bacteria, it is made up of peptide and lysozyme is used, whereas plant cell is made up of cellulose and pectin. So cellulase and pectinase are used to disrupt the cell wall, whereas in the case of fungal cell, it is made up of chitin. Therefore, chitinase is used to release the cell wall. Then RNA is removed using RNase treatment, and protein is removed using protease treatment, and DNA is precipitated using alcohol, ethanol, isopropanol, etc. So fine threads of DNA is obtained, and this can be spooled out and can be purified and can be used for further use. We have already discussed DNA isolation followed by restriction digestion, then agarose-cell electrophoresis or separation of this restricted DNA fragments, and this DNA is eluted and further used for the preparation of recombinant DNA. Then restriction enzyme is used for cutting the DNA, whereas ligase is used for joining the DNA molecule. And finally, amplification of gene of interest using PCR. PCR is another technique which is called as polymerase chain reaction. So it is an in vitro DNA amplification technique. The three steps involved in PCR are denaturation, annealing, and extension. As you see, this double-stranded DNA is made into single strand. That process is called as denaturation. By heating, then primers are annealed. Primers are short strands of DNA that is complementary to this template strand. All DNA polymerase requires a short strand of primer with free 3' OHN for elongating the strand by adding nucleotides. Then the next step is adding DNA polymerase, and deoxynucleotides, DNTPs, are also there, and that causes extension of that particular strand, and this cycle is continued 30 to 35 times. We will be getting billions of copies of a particular DNA fragment. So PCR involves 3 steps: denaturation, annealing, and extension. This makes a PCR cycle. Denaturation at high temperature, double strand becomes single stranded. Annealing, the primer anneals to the single stranded region, and extension by DNA polymerase, which is a thermostable DNA polymerase that elongates by adding nucleotides to the primer. The next is about the DNA polymerase that is used in PCR. So it is a special DNA polymerase. It's a thermostable DNA polymerase that is isolated from Thermus aquaticus, a bacterium that lives in hot springs that can tolerate high temperature. So this enzyme with this thermostable capacity is used in PCR. This amplified fragment can be used for further in recombinant DNA technology. Then insertion of this recombinant DNA into host organisms. We have already discussed making it competent by means of calcium ion treatment or biologistic method. We can introduce that gene. Then the next step is antibiotic resistance gene selection of this recombinant transformed colonies by means of antibiotic resistance gene or by means of beta-galactosidase. We need to select that particular colonies. So we have already discussed that topic. Then finally, obtaining the foreign gene product. So why we are doing all these things? First is we need to multiply the copies of that particular gene fragment. Now we have PCR for that. Then the second is, the most important thing is large-scale production of the gene encoded gene of interest. So suppose this is an insulin gene. So we have incorporated that insulin gene into a bacterium, E. coli. So this gene inside the bacterium undergoes transcription and translation, finally forming the protein product or insulin. That is how humulin or recombinant insulin is produced. E. coli or bacterium acts as biofactories with human insulin gene that produces insulin in large amounts. So maintaining cells in physiologically most active log or exponential phase for the production of large amount of products. So a continuous culture system is used. That means fresh medium is continuously supplied. Then for that we use bioreactors. Bioreactors are large culture vessels for the culturing of desired microorganisms with all optimum conditions. So it provides all optimum conditions for the production of the product or growth of that particular organisms expressing the product. So there are 2 types of bioreactors. First one is simple stirred tank bioreactor and spatched stirred tank bioreactor. So a stirred tank bioreactor is cylindrical and with a curved base where mixing of reactor contents is happening, and the stirring facility actually facilitates mixing of oxygen throughout the bioreactor. Then there is air can be bubbled through this reactor, that is a spatched stirred bioreactor with an agitator system. So for efficient oxygen delivery and with a temperature control system. The final portion of this chapter is downstream processing. What do you mean by downstream processing? So this is a summary of downstream processing. The protein that is produced in bioreactors is separated and purified. The protein must be purified. Then the product is added with certain preservatives or formulations. Suitable preservatives are added to the product. Then this should undergo clinical trials in the case of drugs, in rats, chimpanzees, etc. Before finally releasing that drug into the market, then strict quality control testing for each product before the product is finally released into the market. So we have given videos on almost all topics. If you want further details, you can refer that video. Hope this video is beneficial. Wishing you the very best for your exam once again. Take care, stay blessed. Thank you so much for your support. You are with biologyexamsforyou.com.